want to thank everybody for tuning in and following along in my webinar today. I'm very thankful to have the Flux guys at Fluxion ask me to come over or come log into this webinar and give you guys a little bit of taste of what I'm doing with their BioFlux1000 system. My name is Derek Mormeyer, as you guys can see on your screen, and I am a grad student at the University of Nebraska Medical Center, and I'll just be starting my 4th year there. So today what I'm going to kind of go through here is just a little bit of my research and our lab's research and how we're utilizing this BioFlux1000 system to look at gene expression in Staph Aureus biofilms. I'll begin by giving you guys a little bit of background for people who aren't in particular bacteriologists and are just viewing in just to see the utility of the system. First, I'll start off with some background about Staph Aureus, then I'll get into a little bit more detail about biofilms, how they develop, and then I'll get into the particular operant that we work on in our lab and the functions that we see that them having. And then I'll get into some of the pictures and the videos that I've taken looking at gene expression inside of these Staph Aureus biofilms, and then kind of give an insight into where we're looking to go and how we're going to use this system further on. So to begin with, Staph Aureus, like I previously stated, is it's a big problem in the hospital world where you can oftentimes get these infections that are often mediated by biofilms, as well as it also contains several virulence factors. Staph Aureus is a gram-positive facultative anaerobic bacterium. What this means is that it can grow in both high and low oxygen conditions, and it changes its metabolism in order to actually grow in different spots, making it even a more difficult pathogen to actually eradicate. The particular strain that we're working on in our lab is called UAMS1. For short, I'll be calling it either wild type or U1 throughout the talk. And this particular isolate that we use is a clinical osteomyelitis isolate. Now what that means is that it was actually isolated from a bone infection at the University of Arkansas Medical Center, I believe, a couple years ago. I've included an electron micrograph of a Staph Aureus biofilm just to show you guys and get you, for those people who don't have a feel for what a Staph Aureus biofilm looks like. And you can see that this one's been pseudo-colored, but this is actually a biofilm on synthetic material. And you can see here that it's actually pretty robust, and there's quite a few cells there. Biofilms, as you may or may not know, are simply bacterial communities that are encased within a extracellular matrix. Now, this extracellular matrix can be composed of 3 main components: proteins, polysaccharides, and extracellular DNA, or eDNA, which our lab is particularly interested in looking at, and I'll explain why further on once I get into more details of the talk. But simply put, biofilms are just these bacterial communities with what we previously thought were pretty simple, but are realizing more and more that they're actually more and more complex kind of multicellular organisms, if you have these. So I've included this figure on the right here just to show a diversity of all the different types of biofilm infections that actually can occur in the hospital setting. And when you can see there's 18 different types of infections that you can actually occur, and a lot of them have to do with catheter-related infections, or heart valve, or as you can see over here, pacemaker infections, and even contact lenses can also be a problem. In addition to also having hip and knee replacements that also tend to get biofilm infections and allow these Staph Aureus biofilms to grow and really proliferate and cause further problems inside of the host. In the bottom left here, I've also included a picture or a confocal image of a Staph Aureus biofilm. Now, I just put this in here to get for those of you who don't have a real grasp of what they may look like. So you have this nice mat of cells, and then you get these 3-dimensional structures that actually cause even more problems and allow these cells to become resistant to antibiotics, as well as avert or invade the host immune system. Like I stated previously, biofilm development was previously thought to be a pretty simple process. But what we're learning is that biofilm development is actually a pretty complex process. But here I've kind of simplified it and using this figure to show the basic steps that a biofilm will go through as it grows into these robust structures. Number 1 here, you can see that obviously to begin a biofilm, you need to have a form of attachment, whether that is to a synthetic surface like a knee, hip, catheter, or if it's some inside the host of a human, such as any type of tissue, the cells need to attach some. And then after they attach, they begin to form this extracellular matrix that I previously alluded to. And they start to accumulate into these 3D structures that we call either towers or microcolonies. And then obviously after they go into these large tower structures, they have to disperse themselves so that they can spread the infection and start the whole biofilm process over again. In the bottom panel here, I've included 1 through 5 in a Staph Aureus biofilm, and these are all BioFlux images depicting Staph Aureus biofilm developmental processes. For most of my talk, I'm going to be interested in the development of these large tower structures here, because this is where we constantly see the expression of our two operons that we're particularly interested in our lab. As I previously stated in the last one, we're looking at these two side-inert operons. Now, what we've previously shown in our lab is that these side-inert operons are important for programmed cell death and allowing the release of extracellular DNA into the extracellular matrix, which forms a normal biofilm. As you can see in here in this top schematic, you can see that the side ABC operon is actually regulated by the side R regulator, this was our type regulator. And what we've previously shown is that this side R regulator responds to changes in altered metabolism, central metabolism of the Staph Aureus cell. And in addition, as I'll allude to later in the talk, we've also shown that side R also responds to differing conditions of oxygen. In particular, hypoxic or low oxygen concentrations also induce side ABC expression. Likewise, in the bottom schematic here, you can see that the lyric AB operon is actually regulated by this two-component system known as LIT-SR, where you have a LIT-S, which is a sensor protein, and LIT-R, which is a response regulator. And this has also been shown to have be regulated by different processes in central metabolism. As I previously alluded to in the last slide, we believe that these two operons are vectors of cell death and lysis. And what we propose is that side A is in fact a wholin-like protein. Now, for those of you who don't know what a wholin protein is, wholin proteins are previously thought to be from bacteria, are previously from bacteriophages that actually allow the cells to form these pores in cells and actually lyse the cells open. Now, we believe these are a wholin-like protein and allow these proteins actually get embedded inside the membrane, causing pores and causing cell death. To combat the wholin-like protein to side A, we propose that the LIRG-A is actually an anti-wholin-like protein that actually, essentially plugs up these pores and doesn't allow the cell death processes. As I previously alluded to the last slide as well, we also know that the LIT-SR two-component system actually responds to changes or dissipation in membrane potential. Now, and we've previously shown this with dissipating agents such as GCTP that actually reduces LIRG-A or LIRG expression. And on the left side, there's a brief schematic of basic glucose metabolism as glucose enters the cell and goes through basic glycolysis phenomenon. Now, what I forgot to mention in the last slide is that what we've seen in the side R and side regulon is that, in fact, during excess glucose conditions, as a phenomenon known as overflow metabolism, you have excess glucose, and in fact, the carbon flow is actually directed to different fermentative processes. Different metabolites are formed that actually side R responds to and actually induces side A expression. Now, I won't get too much into the metabolism there, but I just wanted to give you guys a brief, small background of how you think these guys are responding. There's still a lot of steps that we're still currently trying to tease apart in order to understand the full regulation of these processes. It's also well known, remembering the last slide, that a lot of our operons are actually regulated by differences in metabolism. It's also well known that metabolic heterogeneity exists in biofilm development. So what you can see here in this figure that I've included is that throughout you have a lot of different differences in altered microenvironments, where on top you can imagine that you in fact have high oxygen concentration versus the bottom where you have lower oxygen concentrations or anaerobic versus aerobic conditions. And what we're particularly interested in, because our operons are regulated by differences in metabolism, is how these different processes actually affect or how the genes are actually expressed inside of the biofilm during normal biofilm development. And in fact, from a previous study in 2007 by Rainey et al., it's obvious that when they looked at a biofilm microcolony, that the top of the biofilm indeed has higher oxygen concentration than the bottom of the biofilm, where you see a lower concentrations as you go deeper and deeper into the biofilm. So keeping in mind that we have metabolic heterogeneity inside of biofilms, and our two operons are indeed controlled by these differences in metabolic and different switches in metabolism, we hypothesized that the Staph Aureus side and third operons are differentially expressed as a function of the metabolic and physiological heterogeneity found within a developing biofilm. So to first test this, what we did was we grew our cultures in a synchronic state. We looked for hypoxic versus aerobic induction of the side and third operon. Now, for a hypoxic induction in synchronic state, we basically had a larger media to flash ratio, and we grew these cells in a static state, but still not in the biofilm like growth stage. And what you can clearly see here in panel A is that side expression, or the transcript levels using RT-PCR, we in fact in the wild type background have induction of side expression. And likewise, we also see that it remains side R dependent. In addition to this, we also grew wild type with and looked at the LERG-A transcript levels. And what we saw was that LERG-A expression has actually slightly decreased during hypoxic growth, but remains LED-SR dependent. So from this, we're more interested. We want to know where are these operons actually being expressed in the biofilm. So the next step would be we have to find some way to actually track them. So what we did was we generated these transcriptional reporters. And we generated 3 transcriptional reporters that I've included here, and they have and I have a schematic representation of each one. And we have one that's the promoter fusion to LERG-A or LERG-AB, then in the side operon. And additionally, we also included as a positive control an LDH1 promoter fusion. Now, LDH1 stands for lactate dehydrogenase, and it's known to be induced under conditions where there's lower oxygen. So if we want to look for any particular places inside of the biofilm that in fact have lower oxygen, this is a good indicator of exactly where conditions have lower oxygen. So as a proof of concept, what we did was we drew in the same way that we did previously in the last slide for RT-PCR quantification. We drew these reporters, and then we looked using confocal images to identify, in fact, if we're seeing side alert expression. As you can see in the top panel here, I hope, in the wild type background containing the side GFP fusion, we see that in the excess glucose, we indeed, like I previously stated, have induction of the side operon. And also in hypoxic conditions, we see also the induction of the side operon. And in the second panel here, second row, excuse me, you also see that it remains side R dependent. In the side R mutant, we see no induction of side at all. The next 2 rows we see we're looking at the LERG expression in both of the wild type and a LED-SR knockout. And what we see is that we don't indeed see no expression of LERG at any time during these proof of concept and looking at these planktonic cells. And lastly, in the bottom row here, as a positive control, we grew the wild type containing the LDH1 promoter fusion under hypoxic conditions, and we indeed see induction of the GFP fusion. So this is why everybody's here, I'm assuming, is to know a little bit more about the BioFlux1000 microfluidic technology. And I've just included a couple slides in here just to kind of talk about how we use it and the way I go about setting up my biofilm and the real utility of the system and what we're getting out of it. So over here, just a brief description of it is, everybody knows that you can automate this system, so it allows you to take images over an extended period of time, and then you can stack these images and allow these to become movies, which is really nice because I always say this, and I think it's true, it's a cliche thing, but a picture's worth 1,000 words. In a video that's contained of 200 images, it's most likely worth 200,000 words. And I think that's true when you really get to choose apart these biofilms. In panel A here, this is just a basic view of what the system actually looks like. I'm sure many of you guys know this, but just to describe it a little bit, we have the pump here on the right that's connected to tubing to an interface that actually sits on top of your plate. And this plate sits on this automated stage, and it actually can be calibrated and told to connect it to the camera, and you can be told where to take pictures through an extended period of time. In panel B here, it's just a basic cartoon schematic of a 48-well plate. And on the left side, you have all of the input inlet wells we actually add your fresh media and then your output or effluent waste wells, where you actually would flow the media across the cells in this little viewing region here. And panel D kind of correlates to that small viewing region there, where you can see these strictly tiny channels, and then the viewing port here, where actually your biofilms would be have their images taken. And lastly, this in panel C here, you just have this basic schematic of exactly what's going on. It's a side view if you cut out the plate. Just again showing that you have fresh medium on the left side, and then you have your microfluidic channel where you perfuse TSB or EU or your biofilms and allow them to grow over time. And then you have your outlet and your spent media where you can actually collect the effluent and do further analyses from there. For our setup for Staph Aureus in our particular strain, I use an inoculum of 0.8 optical density at a wavelength 600. And what I do is then the cells containing fluorescent reporters are then seeded and then left for an hour to adhere. And then after this hour, I run 50% TSB for 18 hours at 0.6 dynes per centimeter squared, which is equal to 64 microliters per hour. Now, everything in the montage software that actually runs your BioFlux is based on dynes per square centimeter, so you tend to get a little bit weird numbers for your microliters per hour. But after that, while it's running and being perfused with 50% TSB, images are acquired in 5-minute intervals. This is about as fast as you can go for a total plate. Otherwise, you won't have enough time in between images to actually take time points every 5 minutes. In addition, after we have taken all the images, both the bright field and the fluorescent images are then stacked and overlaid and then converted to videos, which I'll show a little bit later. And you can really get a nice feel of how things are growing over in a real-time manner. So I also included a couple kind of BioFlux tips. I know some of these may seem a little kind of childish or kind of, no duh, but the first one here is bubbles are bad for biofilms. Now this sounds funny, but in reality, if you get any bubbles in your system, it really is hard to grow biofilms. It will destroy them, and you'll have to waste the plate, and this is not what you want to do with the system. Additionally, you need to standardize everything before. I've used a particular medium because I standardized it for this particular strain and this particular type of species. Now, what little experience I have with different species of bacteria growing in the system, you can get a lot of differences in growth, and you need to really standardize for your media, your inoculum, and your flow rate. If you don't, you tend to get an overgrowth if you have too high of an inoculum. This actually clogs up your system, and then you don't get a very good biofilm. Or if you don't have enough inoculum to start with, then you tend to not get a very robust biofilm. It's hard to really grasp if you're getting the full potential of the biofilm growth. For better picture resolution, I've included this little snippets down here. I put in here to acquire images manually. If you really want to get a great picture, I know the videos are awesome, and you guys will really show that, but if you want to get a picture for publication purposes, sometimes it's really nice to just take the pictures as a snapshot, and then you can use the videos to supplement that data. Additionally, this sounds like a no-duh thing, but you need to really ensure that the plate, this is basic microscope 101, I realize this, but your interface sometimes can get condensation on it, and any type of condensation can then make your images skewed a little bit, and you can really decrease your picture resolution. Just kind of keep some of these things in mind. And at the end of it, if you guys have any more questions about anything that in terms of the utility or looking at biofilm, I'll be free to answer any of those questions. So anyways, go back to the fun stuff here. So what we did here was start looking at site expression using our transcriptional reporters. And so what we did was we grew wild type and with a side ABC Explorer Fusion. And we grew these guys for 18 hours, as I previously said before. See this confluent layer of cells that begin to form up over time from the basic wild type background. And then there's this a couple minutes here where you actually have this little bit of dispersal. And then you see this beginning tower formation, what we call microcolonies, where we see these very robust structures that actually, in a little bit here, you actually see the delayed expression of side inside the towers. And what this suggests to us, and what this does, it also supports our hypothesis that indeed, inside of these tower formations, it's actually hypoxic, and there's lower oxygen concentrations going on inside these towers. Now, we did the same thing with our wild type containing the LERD AD promoter fusion to GFP. And what you see is you see the very same basic growth characteristics that we previously saw in our other strain background. You see this early confluent layer, and then you see a little bit of dispersal, and then you see this brightly or highly fluorescent constitutive expression of LERD throughout the entire tower formation. And what this suggests to us is that indeed LERD does not respond to conditions that have anything to do with oxygen concentration. We're still starting to pick apart what exactly LERD is sensing here, but it's clear that it's very different from the side expression phenotype that we previously saw in the last video. So our next question that we really wanted to ask was, okay, so we have these towers that are growing, and we see differential expression of the two-side NERD operon. But are these two actually being expressed in the same operon, or excuse me, in the same tower, or are they two different tower types? So what we did was we made a dual reporter transcriptional fusion with a DS red hooked to road AB, and then a side ABC fused to GFP. And what you'll see is a very striking and very similar to what we saw previously. And we see that actually both of the operons are being expressed within the same tower phenotype. So like we previously saw, we see this high level of alert expression constitutively throughout the entire tower. And then you see this delayed expression of side that comes on later on during the developmental process. In addition to this, that I didn't previously talk about, is that we also have this small tower phenotype. I know it may be hard to see on your guys' screens, but I'll show a couple more examples later on in the talk. And what we see is these smaller tower phenotypes that seem to not be able to grow as quickly, and don't divide as rapidly as these large towers that we see constantly during our normal growth period. Now I'm going to play this again just to try to show you guys that we actually do have this other tower phenotype. It'll come up around here. Like I said, if it's hard to see on your screens, I'll show other examples and pictures later on. But what you'll see is that this small tower actually has constant and high levels of side expression. And what it appears to is that it actually is less cohesive than the larger tower phenotype. And it actually looks like they are breaking off and actually could be some other role in terms of biofilm development. And now what we're trying to do is tease apart what these two different biofilm structures are and what their function is in this whole large multicellular community. So to test further if we indeed have hypoxic conditions inside of these towers, we grew our wild type with our LDH1 promoter fusion on the right side here. I wish there was a little bit bigger tower for you guys, but what you can see is that it's clearly inside of these tower phenotypes, or these towers, is that we clearly see that there's expression of LDH1, suggesting that indeed there are hypoxic conditions inside of these developing towers. And on the left, just showing wild type again with the side promoter fusion to GFP, and you see that they both look similar in terms of being inside of these interior regions of these large developing towers. So obviously we know that these two operons are known to be involved in the effectors of cell death. So what our next step was to stain for dead cells in eDNA. Now we use a very, very small, low concentration of ruthenium iodide, and what ruthenium iodide stains, if you may or may not know, is that it stains the dead cells and eDNA because it's an intercalating DNA agent. And so anytime that there's a cell that has a membrane that's compromised, the PI will actually get into the cell and stain that cell, or any eDNA that's found within the biofilm will also be stained. So what we clearly see in the top row here is that in the large towers, and as well as the bottom row here, you see that in the large towers, there's this large staining that's pretty much primarily inside of these towers and is devoid everywhere else. And it correlates very nicely with the cell death and the release of DNA to both of our operons in these particular towers. Additionally, and strikingly and kind of interestingly, that we didn't really expect was that side expression in the small towers is actually completely devoid of the cell death and eDNA release. So this brings up other questions of what exactly these smaller towers, what their function is, and how are they being regulated. So as I previously stated, these are these smaller towers that I was talking about. Now they have this high level side expression in the small towers, and they seem to be, hopefully you guys can see this, these smaller cells that are breaking off and actually less cohesive. And it's interesting that when we ran our wild type in another channel we ran, we see these towers that also look quite similar to the high expressing towers that actually contain the LDH1 expression. Now what this is telling us is that perhaps maybe the different tower types may be in fact linked to altered states of metabolism. So I bring you back to this slide that I previously showed, and what you can see here is that we have these two important enzymes that actually are involved in converting acetyl-CoA and acetyl phosphate to acetate. And what we've previously been shown by a postdoc in our lab is that when you knock these two enzymes out, you actually get an increase inside the site expression inside of the cell. So what our next step was to do is try to figure out, what's still going on. So what we did was we moved our site expression, or our dual reporter, into the ATK and PTA background. And what we see is this very, very high increase in site expression all over the place inside of the biofilms, which is what we would expect. And what we didn't expect was that we'd also see all these smaller tower phenotypes. Now what this is suggesting to us, especially in the PTA, as you can see, and what this is suggesting to us is that these two enzymes are causing these towers to actually grow into smaller structures. And what the function is we still don't know, but what it is telling us is that there's this link to differences in altered metabolism and the expression of the side alpha. So for my talk, there's three main points I kind of want to get across to you guys, just very basic here. So first, there are two distinct tower types, and I think I've gone through them quite a bit, but I just want to point them out and go over them again. First, we have the one large towers. These are rapidly dividing with constitutive large expression, and they also have delayed side expression and an anaerobic interior of the towers. Additionally, we also see this high-level side expressing small towers that appear to be growing less quickly and also seem to be less persistent. The second point I want to make is that cell death in the towers correlate with the side expression. Now, the small side expressing towers are also devoid of these dead cells. It's interesting to note that they may have some other kind of role. And lastly, that the increased number of side expressing towers in the PTA and ATK mutants further suggests a link to overflow metabolism. So with these first kind of primary studies that we've gotten into, it's clear that there's this link to alterations in metabolism. So the next steps that we want to do to generate fluorescent reporters. So what we're going to do is we're going to generate, and actually we're doing this right now, is we'll create these dual reporters that are reporter fusions of the cell death proteins, the cytosine, and also key enzymes of different metabolic pathways. Now, key enzymes of different metabolic pathways, we're looking at enzymes that are basically targets or indicators of which way the carbon in this metabolism is actually going. So we're going to look at different types of enzymes that are indicative of different fermentative processes, also as well as ones that are indicating of the higher states of glycolytic or glycolysis that could be occurring. In addition to this, we're also going to use specialized stains and dyes to determine the metabolic and redox states of the cells. Now, in particular, we're going to use certain stains like CDC, which is actually going to test the redox state of the cells. And in addition, maybe another stain such as HPF, which will also look at these other metabolic states themselves and correlate that nicely and look at where inside these biofilms they're indeed being expressed and what is the metabolic state of the cell at that particular time. And thirdly, one of the other things that we plan to do is I've recently figured out how to get the biofilm actually out of the channels nicely. Is what we're going to try to do is also isolate RNA from the cytosine expressing cell. And what we're trying to do is once we get this fluorescent cells out, we want to take and do RT-PCR and to have specific probes for metabolic pathways that are active during these cells to get a better understanding of indeed where these carbon flow is actually going through. So with that, I may have gone a little fast, but I want to acknowledge everybody in the Bales Lab. We got a big group, and we got a bunch of people working on these outruns, and it's really been a good time. I want to thank my supervisor committee for all their advice and their input on my project. And I want also give a shout out to Ethan Mann for making the promoter fusion, and then also Kelly Rice at the University of Florida. And lastly, I want to give a special thanks to everybody at Fluorescent for allowing me to do this webinar. It's really a treat for me. It's a new experience that as a grad student, I know a lot of us get to do this, so it's really kind of nice for everybody to invite me there. I also want to have a special thanks to Brian and John for talking to them at the ASM conference, and they asked me to do it. It's nice to share, and here we are now. And also, I want to thank Ollie for heading up everything with the webinar and really giving me everything I need to know to work the system. So with that, I'll be glad to take any questions. I have enough time here, and we can take some questions. Also, if I can't get to your questions today, feel free to email me, and I'll definitely get a shot at trying to answer your questions, especially if it has to do with anything in terms of biofilm, or if it's just a question on something on how to use the system, or you want an unbiased opinion on the system, something along those lines. So thank you very much.